Okay. Um, in this podcast, we want to address um, the foundations of chemistry because this is the basis of a lot of the physiology that you'll be learning in the course. Okay. And I know that chemistry can be a complex subject, but what we're going to try and do is break it down to the point where what you learn about it is useful in the context of this course. And obviously, any questions you have, feel free to ask me. So, in order to talk about chemistry, we kind of have to pull back and go all the way to the beginning of time. Okay? So it's a very, very long time ago, billions of years ago. Okay? And at some point, a long time ago, all the matter and energy in the universe was concentrated in a single tiny point called a singularity. Okay? It's what physicists called it. And that created an unstable situation that led to a giant explosion known as the Big Bang. And when the Big Bang was created, the stuff of the universe basically came into existence. And those three things are matter, energy, and empty space. Okay? Empty space is empty space. There's nothing there. Okay? Energy is the ability to do work, and it comes in a lot of different forms, as we'll see in just a minute. And then matter is anything that occupies mass or occupies space and has mass. And there's three kinds of matter: solid, liquid, and gas, that vary according to the amount of movement that the three states of matter have. Okay, in a solid, we have a definite shape and a volume, and the positions of the atoms or the molecules that make up the solid don't change over time. Okay, that's why it tends to retain its shape and its volume. A liquid has some degree of movement in that phase of matter, and that's why a liquid can form to the shape of its container. Okay, and a gas is a type of matter that has even more movement associated with it, and it touches all the walls of the container that it's in, such as the air in the room you're sitting in right now. Okay, you can change from one phase of matter to another by changing conditions like temperature and pressure. Okay, um, every known substance can exist in any of these three states, depending on the conditions. And you can even draw a diagram where that happens called a phase diagram that is specific to a particular element or a particular molecule. Okay. Now, energy is just the ability to do work. Okay. And energy can change from one type to another. Okay. An example is here in this little diagram, right? You can see this gentleman
Energy is the capacity to
pulling
perform
back.
work. Potential energy is energy that is stored. Kinetic energy is the energy of motion. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy. The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
So you can see that energy transformations happen all the time, and every time they occur, some heat is given off, and what that heat does is it randomizes the universe, so it makes the arrangement of matter more random, and as a result, more low energy. Okay, so examples of energy transformations that you might be familiar with, if you've ever gone to a junkyard and see a car picked up by a giant electromagnet and dropped into a car crusher, that's another example of energy transformation. You can turn electrical energy, which is the movement of negatively charged particles called electrons around a large hunk of metal like an iron disc, into magnetic energy, and that's called electromagnetic radiation. Okay, and that is used to pick up the car. Right, the car rises from where it was sitting. Okay, and so that's an example of the electromagnetic energy causing causing kinetic energy to take place, and then the car now has more potential energy because it's now above the ground instead of lying on the ground, right? And then when you drop the car into the crusher, okay, you cut the electricity to the electromagnet, the magnetic field disappears. Now there's no energy holding the car to the magnet, and the potential energy of the car, which was up there with the magnet, becomes kinetic energy, and the car falls into the crusher. And in every transformation, some heat is given off. Okay. Now, there's two rules that control the universe when it was created, and those are called the first and second laws of thermodynamics. And they're pretty simple, right? The first law of thermodynamics says that energy and matter can't be created or destroyed, so there's only so much stuff in the universe, and that was created at the Big Bang, and we can't wish something into non-existence or make something appear out of nowhere. Okay, there's no magic. And the second law of thermodynamics states that if we consider any system, all right, like the air in the room or the water in a glass or the oil and vinegar and spices and water in a bottle of Italian salad dressing, okay, that if we don't put any energy into the system will try and arrange itself in the lowest energy state because that's the easiest thing to do, okay. And we see this all the time when you, if you've ever seen somebody blow a smoke ring, right, with a cigar, okay, that smoke ring initially will maintain its shape, right, but if you wait long enough, it will diffuse into the air in the room, and the air in the building, and the air in the city, the air in the state, the air in the country, then the air in the world, and then eventually it'll drift off into space. And that didn't require any energy to happen. That was driven by the tendency of all systems to try and find the low energy or most random state. Okay, and we'll see this as a theme over and over again when we talk about chemistry. Okay, energy can be converted from one type to another, and at every conversion, some of that is lost as heat, and that makes the universe more random. Okay, and that's part of what maximizes the entropy of the universe. Okay, matter is essentially made up of members that are called elements. Okay, an element is something that can't be broken down into a simpler substance by chemical methods. Each element has unique physical and chemical properties. Okay, the physical properties are things that we can pick up with instrumentation or with our senses, such as the fact that some metals are Um, Malleable, meaning they could be bent. They conduct heat and electricity very well in many cases. Okay. Um, some elements are denser than others. You know, a lead brick is of a particular volume is much heavier than an aluminum brick of the same volume. Okay, that's a property called density, which is mass per unit volume. Okay, and then the chemical properties are basically the way that the element interacts with other elements. Okay, so how do they buddy up or come apart when they're in close proximity to each other? Okay? Atoms are the tiniest building blocks of elements, and they're basically the smallest unit of matter that maintains the chemical and physical properties of that element. Okay, and it comes from an ancient Greek word meaning a, not, and tomos, divisible. This was a thought experiment that was performed by an ancient Greek named Democritus. One day he was staring at a boulder, and he thought if he split that boulder in half, he'd have two smaller boulders, but they'd still have the basic properties of the original. And if he continued the process, he'd have four even smaller boulders that would still retain the properties of the original. But he reasoned that if he kept going, he would eventually get to a point where he could no longer divide those components into something smaller that retained those properties, and he called that particle the atom, which is essentially the same definition we have today. Okay? Atomic symbols are one or two letter chemical shorthand for each element. We'll show you an example of that in a minute. The model of the atom that we currently work with has a nucleus that contains particles called protons and neutrons that have most of the mass of the atom. And then outside the nucleus are electrons that move around the nucleus at a particular distance. Okay? The protons have a positive electrical charge, the neutrons have no electrical charge, and the electrons have a negative electrical charge. Okay? The electrons move at the fastest speed anything can move, which is the speed of light. All right, and they're always found at in these particular locations outside the nucleus, which are called orbitals. Okay, um, the planetary model has them orbiting around the nucleus like planets around the sun, and the orbital model has them in a three-dimensional distribution around the nucleus. Okay, um, the model of the atom that you're looking at here obviously has some flaws in it. The electrons with a negative charge and the nucleus with a positive charge should attract each other, and so you might reason that the atom should either collapse or explode, right? Because opposite charges attract and similar charges repel. But the atom doesn't do that, okay? And you might wonder, well, why is that? And the answer that comes to us from many years of research is we don't know, okay? A lot of times you don't get satisfactory answers even from lots and lots of time spent researching a phenomenon. We're still learning about the atom. But what we do with what we know about the atom is we generate something called a model, okay? And a model is just our best guess at what we know at the time. And models change all the time. Models need to be thrown away when new knowledge comes to light, okay? And then they get modified. But the purpose of a model is just to take what the model gives us, and if it's useful, we sort of hold on to the model as a tool, okay, in order to try and understand more about the world around us, okay? So just keep in mind, models are not necessarily the situation at hand. They're just our best reasonable guess, okay? Different elements have different numbers of subatomic particles. Example, hydrogen has one proton, no neutrons, and one electron. Lithium has three protons, four neutrons, and three electrons. And helium has two electrons, two protons, and two neutrons. Okay. Um, okay. You can see that these atoms arranged the way that they are, right? With most of the mass in the nucleus, have electrons outside the nucleus that exist in particular orbitals. Okay, so what determines what goes where? Well, um, the first orbital, which is the one closest to the nucleus and the one with the least potential energy, so it has the lowest energy electrons in it, can hold a maximum of 2 electrons. The next orbital can hold a maximum of 8, okay? And then the orbitals beyond that can hold even more, okay? Now you might ask why. And the answer again is we don't know. It just is, okay? We take what the model gives us and we move on. It turns out, okay, that if we look at all the elements in the universe, we can summarize them by the periodic table. Okay, this is a periodic table of the elements that shows you some different colors indicating different things. This stair-step area over here divides the non-metals from everything to the left over here, which is a metal. Okay, and then this last column over here are called the noble elements because they don't do any chemistry. They don't make or break chemical connections with other atoms, and they're found in their pure states in nature. So that indicates that they're in a low energy state. Since they don't do any chemistry, they must be quote unquote happy the way they are. They're in that zone described by the second law of thermodynamics that says if we consider a system like a container of neon or helium, that it will, if we don't put any energy into it, adopt its low energy state. Well, the low energy state for these guys is just to be left the way they are. Okay, they don't bond with anything, they're just found the way they are. And so that should tell us something about the other elements. Okay, the other elements all have incomplete outer shells. Okay, so they're either going to attempt to gain or lose or share electrons when they do chemical reactions, which is nothing but the making and breaking of chemical bonds, in order to try and get to the point where the outer shell is filled up so that they're in a low energy state like these guys in the last column. Okay, the columns are called groups and the rows are called periods. Okay, the lightest stuff is in the upper left hand corner, the heaviest stuff is in the lower right hand corner. Okay, um, when non-metals react with each other, okay, when they form connections with other nonmetals, they generate something called covalent bonds, where electrons are shared between the elements. When metals react with nonmetals, okay, such as the formation of sodium hydroxide would be one example, right? Sodium is a metal, oxygen is a nonmetal, and hydrogen can be considered a metal or a non-metal, depending. Okay, they form what are called ionic bonds, and in an ionic bond, electrons are lost by one element and gained by another, and that causes the charges on those atoms to change, so they have either a net positive or a negative charge, and it's the opposite charges that hold the ionic bond together. Okay, metals tend to be shiny. They tend to conduct heat and electricity very well. They tend to be malleable. And in chemical reactions with nonmetals, they tend to lose electrons. Okay? Nonmetals are poor electrical and thermal conductors, and when they react with metals in chemical reactions, they tend to gain electrons. Okay? An atom that loses an electron is called a cation. Okay, so it has a positive charge, and an atom that gains electrons is called an anion, so it has a negative charge. The way I remember that is that the T in cation looks like a plus symbol. Okay, the numbers at the top of each of these columns, all right, indicate the group designation. This is group 1A, 2A, 3A, 4A, and so on. Okay, you might wonder, before we move on, what kind of bonds form when metals react with metals. Those are called metallic bonds, and they're beyond the scope of this course. Okay, this is one of the ways, for instance, that you form metal alloys. All right. The numbers at the top of each column just indicate the number of electrons in the outer shell of each of these elements. All right, so everybody in this column has 1 in the outer shell, everybody here has 2, everybody here has 3, everybody here 4, 5, 6, 7, and everybody here has a complete outer shell. Okay, so Uh, inside the box, you see a one-letter symbol that indicates the identity of the element, like boron, carbon, nitrogen, oxygen, chlorine, bromine, chromium, lithium, sodium, and so on. Okay, and then at the top, you see the atomic number, which is always a nice round number. The atomic number is equal to the number of protons in the nucleus of the atom, which in a normal atom also happens to be equal to the number of electrons orbiting the nucleus. And so all atoms are electrically neutral. Okay? And then the number underneath on the bottom, the bigger number, is the atomic mass. Okay? And the atomic mass is just the sum of the protons and the neutrons in the nucleus along with the electrons, which have virtually no weight. Okay? So for instance, oxygen with an atomic mass of 16 has 8 protons and 8 neutrons in the nucleus. Okay? You know there's 8 protons because of the atomic number, and the rest of the mass is made up of neutrons. It also happens to have 8 electrons orbiting the nucleus, with 2 in the first shell and 6 in the outer shell. Okay? The purpose of chemistry, right, is to describe the interactions between the other elements that want to buddy up in an effort to have a low energy state, right, to have a complete outer shell of electrons. And they do that by gaining or losing or sharing electrons. Okay? And the reason that they do this is because all the other elements besides the ones in this last column have these incomplete outer shells. Okay? You can see some examples of scientific notation to describe atoms here, right? Atomic mass, atomic number, mass on the top, number on the bottom, and the symbol to the right. So that's the shorthand that you would see if you were writing this in your notes or if you saw it in a textbook. Okay? So the atomic mass, right, the number of protons and neutrons. The atomic number is just the number of protons and is always unique for each element. Okay? Isotopes are just versions of atoms that have a few extra neutrons in them. Okay? They have the same atomic number but a different atomic mass. Some examples of isotopes of carbon are shown here. You can see that the atomic number is the same in each case, but the atomic mass is different. Okay? Carbon 14 is heavier than carbon 12, but if I were to go out and collect carbon atoms, right, most of them would be this version of the carbon atom, which we would call the most abundant isotope, but some of them would be these heavier versions of the carbon atom, which are less abundant isotopes. Okay? Isotopes, in some cases, give off energy that we can measure. All right? In some cases, it's electrons that get ejected from the isotope. In some cases, it's X-rays. In some cases, it's alpha particles, which are basically helium nucleus, right, with 2 protons and 2 neutrons. It all depends on the isotope that we're talking about. In many cases, this energy that gets given off results in something called decay, where the radioactive isotope slowly changes into another element because the composition of the nucleus changes. All right? But the reason we care about isotopes in anatomy and physiology and in medicine is because we can use the energy given off by these radioactive isotopes to ask questions about what goes on inside the body. Okay? For instance, X-rays can be used to visualize broken bones, and we can use contrast dyes to visualize things like blood vessels or the flow of liquid through the tubules in the kidney, for instance. We can use radioactive sugar that contains some carbon-14 in order to trace the use of sugar in the brain using a detector that can pick up where the sugar goes. All right? And that's called a PET scan. So these techniques are important because they allow us to look inside the human body without having to cut you open and sort of mess around trying to find the problem. All right. In the old days, before we had this kind of material and these techniques at our disposal, we used to have to do exploratory surgery in which we would open you up and look around inside to try and find the problem you were complaining about. If we couldn't find it, we would sew you back up and tell you to hope for the best. And basically, you had to come back in when the problem reoccurred. And sometimes they found the problem, sometimes they didn't. Okay, but now that we can look inside the body and locate exactly where damage is located, surgery is shorter, more likely to be successful, and results in shorter times in the hospital. Okay, so it was a big step forward. Okay? Ions are just atoms that have lost or gained electrons. Okay? Cations are positively charged and anions are negatively charged. There are ions in the human body, and the reason that we care about them is because they are charged particles, and when charged particles move, that is a form of energy called current, and that can be used to do work. Just like current in a wire is the movement of electrons from one place to another. It can be used to turn on an electric light or an electric motor. Okay, current in the body is the movement of charged particles from one location to another that can be used to do work. Nerve cells use it to send electrical signals from one place to another, and muscle cells use it to generate muscle contractions, and every cell in the body uses it to help transport material across the barrier known as the cell membrane. Okay? The other thing that electrolytes do is they help us control where water goes in the body through a process called osmosis. Okay? There's a property of passive transport, which is the movement of molecules or ions from an area of high to low concentration that governs everything in a liquid or a gas. All right, and the rule basically states that where the ions go, the water will follow. Okay, so we can use the ions in a liquid to pull water into a particular location through osmosis. Okay? The atom is happy when it has that full outer shell, right? We should say, it says all other shells can hold 8 electrons. The second shell can hold 8. The third shell is happy with 8, but can go up to, I believe it's 17. All right. But the point of a full outer shell is that once the shell is full, you don't have to do any more chemistry. So you form what's called a stable molecule. Okay. The fact that the 8 electron rule holds for the second and third shells is called the octet rule, meaning we try to get that 8 electron count in that outer shell so that everybody in the molecule is happy. Okay, octet means 8. For the first shell, it's called the duet rule. Okay, 2 electrons is a full outer shell in that case. Okay, some examples of different atoms are shown here. Right, hydrogen, carbon, nitrogen. Notice that in every case, we have an incomplete outer shell, right? Hydrogen with 1 can pick up 1 to make 2. Carbon with 4 can pick up 4 to make 8. Nitrogen with 5 can pick up 3 to make 8. Oxygen with 6 can pick up 2 to make 8. Okay? Phosphorus and sulfur are examples of phosphorus is a sulfur is a non-metal, okay, and phosphorus is a non-metal, and basically what they will do is pick up electrons to try and complete their outer shell. If it's with another non-metal, it'll be through sharing, and if it's with a metal, it'll be by picking up electrons from the metal. Okay, so notice here, right, the different arrangements of the electrons and how they always obey this distribution, just like floors in a hotel, right? The first floor of the hotel atom can sleep 2, the second floor of the hotel atom can sleep 8, the third floor of the hotel atom can go up to 17, but is happy with 8. Okay. So one type of chemical bond that can form is called the ionic bond. And in an ionic bond, the non-metal picks up electrons from the metal. They take on opposite charges, and that's what holds them together, just like opposite poles of a magnet. Okay? An example of an ionic reaction is shown here, right? Sodium is a metal, okay? And if I were to bring pure sodium into school, I would have to bring it in under gasoline because it reacts so violently with the water in the air that it bursts into flames, okay? So it's an extremely dangerous element, right? Chlorine is the kind of stuff that'll bleach your skin and your clothes, and it's extremely caustic. All right, if you've ever been around a pool when it's being chlorinated, you know that it can burn your eyes and your skin. And when you mix these two elements together, what happens is that the sodium loses its outer shell electron, and that makes it form a cation that has a complete outer shell, so it's stable, right? Well, the chlorine atom picks up the electron from the sodium and adopts a negative electrical charge and completes its outer shell for that stable configuration of 8. And what holds them together are the opposite charges, okay? And this forms sodium chloride, which is table salt, which is what you put on your French fries, okay? So how is it that you can take two extremely dangerous elements, put them together, and make something that you can eat? And the answer is you've changed where the electrons are located, and when you do that, you change the chemical properties of the resulting molecule, all right? If you were to look at a crystal of sodium chloride, you would find that the chloride anions are interspersed with the sodium cations in a way where that they alternate, right? And what holds them together are their opposite charges. And the reason for that arrangement is it's low energy, right? It's real easy to have opposite charges hug each other and then stack all these things the way they're shown here. If I tried to force all the sodiums to one side of the cube and all the chlorides to the other side, I could do that, but it would take a lot of energy to maintain because the similar charges would push on each other, all right? So this is the low energy state. This is the second law of thermodynamics at work. If I were to take that sodium chloride crystal and drop it in a glass of water, all right, the water, as we're going to find out, has a positive and negative end to it. And so the water kind of tries to shove itself between the sodium and the chloride, and when it does that, it forms a little cage of water molecules around the ion, and the ion falls away from the crystal, and the crystal eventually dissolves when all the ions are what we call hydrated, that is, surrounded by water molecules. Okay, and y'all know this. Y'all know that salt dissolves in water. You can continue to do that up to a point, right? Eventually, you get to a point where if you keep adding salt to a certain volume of water, it won't dissolve anymore, and that's called the saturation point. And the reason that happens is that you run out of water molecules to dissolve the crystal, okay? So in a covalent bond, these are chemical reactions that occur between nonmetals, okay? That's to the right of the staircase and to the left of that last column in the periodic table. And basically, this is the sharing of electrons between two elements that holds them together. And by sharing, both atoms get to count those electrons in their outer shell, and that results in a stable molecule, right? So notice here, right, the hydrogen molecule, 2 hydrogen atoms, 2 electrons in the outer shell for that hydrogen, 2 for that hydrogen, stable compound, full shell, right? Oxygen, let's count. 1, 2, 3, 4, 5, 6, 7, 8 for that oxygen, right? And then 4, 5, 6, 7, 8 for this oxygen. And so that's a stable compound. In this case, they're sharing 2 pairs of electrons, so they form what's called a double covalent bond. And that's going to be stronger than a single covalent bond. It's like a tougher cable, okay? These are molecules because they're made up of identical elements. This is a compound, okay? This is methane. Different atoms are participating here. But if we do the bookkeeping here for methane, you can see there's 2 electrons for this hydrogen, 2 for this one, 2 for this one, 2 for this one. And then for the carbon here, we got 2, 4, 6, 8. So we have complete outer shells for all the elements. So we form a stable compound, and the result is that that thing will hold together. All right? So single bond, double bond, and you can even form triple bonds in some cases. Okay? Example of that is between something like carbon and nitrogen. All right? So you can see here the formation of water molecule. Right? This is another compound. Right? An oxygen and two hydrogens come together. Right? Two electrons for that hydrogen, two for that hydrogen, and eight for the oxygen. All right. Now, an important point about the elements to the right of carbon and to the left of that last column in the periodic table is that they have a property called electronegativity, meaning that those elements, things like oxygen, nitrogen, sulfur, chlorine, fluorine, iodine, and so on, they tend to have a greater pull within a covalent bond than something like carbon does. Okay? And the result is that the electrons spend a little bit more time with the electronegative atom than they do with the atom that is less electronegative, or we could say more electropositive, right? So you can draw some different models of what this looks like when the molecule forms, okay? The orbital model here is showing you the distribution of electrons where most of it's going to be around the oxygen, so there's a slight negative charge around the oxygen atom and slight positive charges around the hydrogens. And the space filling model looks like this, okay? So this is a condition in which there is a separation of charge, and we call those molecules polar, all right? And the second law of thermodynamics simply states that in this situation, polar things like to be with polar things or charged things. And the reason for that is that they can associate by having their charges alternate, right? The negative part of one molecule you get with the positive part of another, and that means they mix real easy, okay? Nonpolar molecules, things like methane and a lot of hydrocarbons like oil and fat, okay, tend to have an even distribution of electrons over their surface, and as a result, they don't associate very easily with things that are polar, okay? So the rule to remember here is like dissolves like, okay? Things of similar chemical properties like to buddy up in a low energy situation. You can see this, for instance, in a bottle of Italian salad dressing, right? When you pull it off the shelf, the oil's on the top and everything else is underneath. The stuff underneath are the water, the spices, the salt, and so on. The stuff on the top is the oil. All right. If you take that bottle and you shake it, you add kinetic energy to it. You form a temporary mixture called an emulsion. All right. But if you let that system sit for a second, the oil will separate and everything else will settle to the bottom, and that's the low energy state. Okay. So that tells you why these properties are important when we consider chemistry. Okay? This is also why water is sometimes referred to as the universal solvent, all right? Because charged things and polar things dissolve in water, and chemical reactions only happen when there's movement going on, right? So that would be in a gas or in a liquid. So in a water situation, you've got stuff moving, which means stuff can bump into each other, which means that can make and break chemical bonds. That's how a chemical reaction happens. Is it two molecules or an element in the molecule or two elements bump into each other, and that energy is enough to crack some chemical bonds and make new ones. Okay? So you can see here that in the case of a polar molecule that has hydrogen atoms in it, the partial positive charge on the hydrogen atom and the partial negative charge on another uh, electronegative portion of a molecule, in this case could be another water molecule, would be an example, will form a temporary attraction, which is called hydrogen bonding. Okay, so that's an attractive force between the electropositive hydrogen of one molecule and an electronegative atom of another. Could be an oxygen, could be a nitrogen, could be a sulfur. Okay, this happens usually in the context of uh, organic molecules, but water is an inorganic molecule and also does hydrogen bonding. It's not considered a true bond because it's between molecules or between regions of large molecules, all right? And it's temporary, but when you add up the net effect, it can have a significant impact on the properties of that particular compound, all right? So let's take a look at hydrogen bonding.
The atoms that make up a water molecule are in a constant tug-of-war over their shared electrons. Oxygen exerts a far stronger pull on the shared electrons than does hydrogen, and so the electrons spend more of their time closer to the oxygen atom. Because of this unequal sharing of electrons, the oxygen atom in a water molecule actually has a slight negative charge, and each hydrogen atom has a slight positive charge, even though the water molecule as a whole is neutral. Because of the unequal sharing of electrons and the resulting positive and negative poles, a water molecule is said to be polar. The polarity of water molecules causes them to be attracted to each other. Since the positively charged atom involved in this special type of attraction is always a hydrogen atom, this kind of bond between molecules is called a hydrogen bond. Each water molecule can hydrogen bond to 4 other water molecules. A hydrogen bond is weak and lasts only a tiny fraction of a second, but it takes a lot of energy to overcome the combined attraction of many hydrogen bonds. This explains water's great capacity to store heat, its high boiling point, surface tension, and several other unusual properties.
So you can see an example of the attraction here. The water molecules move past each other and they flip and flop in order to get the partial positive regions of one water molecule near the partial negatives of another. The properties this imparts to water include a high heat of fusion and vaporization, meaning that water can absorb or give off a lot of heat without changing temperature very much because of the attraction of hydrogen bonds. Surface tension, which is the ability of water to kind of stick to itself. That's why water can wick up a paper towel or get from the roots of a tree all the way up through the trunk into the shoots. Okay, um, and it's also why when water changes from a liquid to a solid, it expands. Okay, you have to maintain that spacing in order to satisfy the second law of thermodynamics. And when we go from a liquid to a solid, that spacing makes the ice less dense, and that's why ice cubes float in a glass of water. Okay, if you've ever been absent-minded and left, say, a six-pack of Coke or beer in the trunk of your car in January when it's real cold, and come out the next day and you open the trunk, you can have a bunch of split cans. And why is that? Because the water in the beverage froze, it expanded and split the can open. Okay, that's also why cracks in your basement or in the sidewalk get bigger in the wintertime, because as the water in the crack melts and freezes, when it freezes, it expands and it makes the crack bigger. Okay, um, the hydrogen bonding between water molecules can be broken by agents like detergents. Okay, so uh, an example would be you've ever seen those insects that can walk across the water, called water striders, right? If you put soap in the water, they'll sink because you've broken the hydrogen bonding, and the water strider is using the hydrogen bonding to kind of hold its weight up, right? It's also part of what makes a duck float, right? A duck floats on water for several reasons, right? One is the waxy coating on its feathers, which repels the water. Another is the fact that the lungs in the bird hold air, right, which is lighter than the water. And then the other is the surface tension of the water. So if you put detergent in the water, the duck will sink because the waxy coating on the feathers will be broken up by the soap, as will the surface tension, and the duck will go down. Okay, I'm not recommending that any of you go out and do that, but that would happen. Okay, and that's an example of the power of hydrogen bonding. Okay, so we're going to move now from the atomic level and we're going to move up to the molecular level. All right, we know already that you can form covalent bonds and ionic bonds, and you can generate connections between atoms that can be small molecules or big ones, or small compounds or big ones, right? Compounds are made up of different atoms associating with each other. Molecules are the same atom, all right? But an interesting property of carbon, right, this guy here, is that he can form rings and chains with himself, and as a result, carbon is the basis of all life, because with carbon, you have the ultimate Lego piece, right? You can build all kinds of interesting things with carbon, including the tools of life, which are called proteins, okay? Carbon's also found in carbohydrates, which you know better as sugars. We'll talk about those in a second. And it's also found in lipids, which you know better as fats, okay? The energy in the carbon-hydrogen bond, which you can see here, right, is significant. And when those bonds are cracked, the energy that comes out can be captured by living systems and then transferred to a molecule called ATP. And ATP is like biological gas in the tank. It's a form of energy that can immediately be used to do work for the cell or for the tissue. Okay? It's a little bit like how gasoline in your car makes your car go, right? Um, think about your car, right? Your car, if it's sitting at the bottom of a hill, okay, would not roll up the hill all by itself. All right. In order to go up the hill, what do you have to do? You have to put the key in the ignition. You have to release the parking brake, and then you have to step on the accelerator pedal. The spark plug then causes an explosion inside the engine block, which fills with gasoline and air. That makes an explosion. The pistons in the engine block move, and that turns the crankshaft, and that eventually turns the wheels of your car, and you go up the hill. Okay? So that's an example of chemical energy in the gasoline. When it explodes because of a spark by the spark plug, that turns into heat and pressure, right? And that can be converted into kinetic energy, moving the pistons and then the camshaft and then the wheels, and you lose some heat at every step, right? And then you go up the hill, okay? So what you're doing is you're harnessing a reaction that is favorable, right? The explosion of gasoline, that's something that wants to happen, to a set of conditions that won't happen on their own, the car rolling up the hill, all right? Well, your car's gasoline comes from crude oil, which is a sticky black liquid that comes out of the ground. And if you were to put crude oil directly in your gas tank, your car wouldn't run. And the reason is that the the components of the engine block and the spark plug and everything else aren't set up to take the energy out of crude oil and turn that into car movement. Okay, all you do is gunk up your engine. But if we take crude oil and we crack it into tinier pieces, we generate a molecule called octane. Okay, octane is an 8 carbon molecule. Octo means 8, and the octane is the component in gasoline that makes your car go. Right, when you mix octane with oxygen and a spark, you generate an explosion, and you give off water and carbon dioxide, and then the energy from the explosion is harnessed to ultimately turn the wheels of your car. Okay? Another important principle this illustrates is that you can use the energy from a reaction, chemical reaction that gives energy off, in order to do work that requires energy to happen. And in the body, reactions that give energy off are called catabolic reactions, and reactions that make stuff and require energy are called anabolic reactions. So the chemical reactions that create you, all right, need the energy from the chemical bonds in the food you eat. And when we crack those chemical bonds in the food you eat, steal that energy, give it to ATP, and then take it from ATP to do work, all right, that's the anabolic reactions driving the catabolic reactions. The sum of all those reactions together is called metabolism. Okay? An example of an organic molecule important in life are carbohydrates. Okay? Sugars can exist in a single ring form or a chain of multiple rings. All right? Examples of chains of sugars, monosaccharides, mono meaning one, are shown here. Okay, this is a molecule that's called cellulose and glycogen, right? Glycogen underneath and cellulose on the top, right? Um, Carbohydrates are primarily used for energy in the body and in some cases for energy storage. Okay. There are carbon hydrogen bonds in carbohydrates. You can see them here. Okay. And when we crack those, the energy that comes out can be given to ATP and used to do work. Okay. The carbohydrate that we eat from plants that we can digest and use the energy from is called starch. The carbohydrate in plant that we can't break down is called cellulose, and you know that better as fiber. Okay, we'll see a picture of that in the next page. Fiber just passes through our body unprocessed, and the reason that we can't get the chemical energy out of fiber is because we don't have the enzymes to crack down the fiber molecule into its simplest pieces, which are called the monosaccharides. All right. Let's look at a little bit of information regarding monosaccharides and polysaccharides here.
Carbohydrates are made of carbon, hydrogen, and oxygen atoms arranged in various ring structures. The disaccharide sucrose is formed in a dehydration synthesis involving the monosaccharides glucose and fructose. When two monomers combine to form a dimer, a water molecule is produced from the dehydration synthesis reaction. Notice which parts of the two monomers were involved in forming the sucrose. The glucose, OH, that gave up its H is the one on the carbon next to the ring oxygen atom when you move clockwise from the oxygen atom. The OH lost from fructose is on the carbon next to the ring oxygen atom when you move counterclockwise from the oxygen atom. When a complex carbohydrate breaks down to simple sugars via hydrolysis, a water molecule is split between the two polymer subunits.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch, in a sprouting seed or in the intestine of an animal, produces this disaccharide. Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay, this is another polysaccharide. This is cellulose. You know this better as wood. This is the fiber in our diet. Okay, this passes through our digestive system without being absorbed into the blood, and it's an important part of our diet because it adds bulk to our stool, to our poop, right? It does that by drawing water into our poop from the fluids in our body, and it makes it easier to pass, okay? Cellulose is basically the structural part of plants. It's kind of like their skeleton. And because it's polar, it attracts water. And that's how we're able to get water from the roots to the shoots in a plant. And that's why a plant can grow real tall, right? The water is attracted to the cellulose. The water is also attracted to the other water. So as the water wicks into the cellulose, the other water molecules follow it. And that allows us to get all the way to the leaves on the plant. That's why plants can grow very, very tall, right? Is that attraction. Proteins are the workhorses of living systems, okay? They contain carbon, hydrogen, oxygen, and nitrogen, as well as occasionally some sulfur and some phosphorus. Proteins are polymers of building blocks that are called amino acids. All right, there's 20 amino acids that are joined together by covalent bonds when we build a protein. Covalent bond between two different amino acids is called a peptide bond, and it's always formed between the amine group of one amino acid and the acid group of the other. Okay, we'll see how they're built in just a second. Right, they can act as either an acid or a base depending on the acidity or the basicity of the liquid that they're in. Okay, acids basically have an excess of protons, and bases have fewer protons than a substance that's considered neutral, like water, which has a pH of 7. Okay, and all amino acids are identical with the exception of what we call the R group. Okay, examples of the different groups of amino acids are shown here. Right, they fall in 3 classes: charged, polar, and nonpolar. Charged amino acids have the basic amino acid backbone, which you can see here. Okay, I'll circle it for you so it's easier to pick up. Let's do white. No, let's do black. Okay. So the amino acid part, right, is this part here. And then the R group is this part that's hanging off. So I'll circle the R group in a different color. Okay. Let's do green. Okay, this would be the R group here. And the reason that this is in the charged group is that this collection of atoms, a carbon here, a double bond to an oxygen, and a single bond to an OH, is called a carboxylic acid group. And acids are defined as substances that in water tend to lose protons to the water. And when that happens, the pH goes down, which means the acidity goes up. Okay. Over here, this is glutamate, right? So the R group of glutamate is this guy. Okay. He is another negatively charged amino acid, right? And then the amino acid part is this part here that I will circle in black. Okay. So again, the reason it's called an amino acid is because there's an amino group, the NH2 here, coming off this carbon, and there's a carboxylic acid group over here. The R group is this group of atoms that can be different from amino acid to amino acid. Okay, we can group the amino acids into charged, polar, or nonpolar. Charged amino acids have a positive or negative charge at neutral pH. Polar amino acids have electronegative atoms in their R group, such as sulfur here. Examples here, like in proline, you've got nitrogen, you've got oxygen here in threonine, and over here in serine you got one, and then over here in tyrosine you've got one, and you've got a sulfur over here in methionine. Okay, so these are polar amino acids. Glycine is also considered polar because it's just got hydrogen as its R group. Okay, and then nonpolar amino acids have mostly carbon-hydrogen R groups, right? Such as the phenylalanine here. This is the phenyl group. These are all carbons here at each bend, right? And hydrogens attached, right? These are double bonds here in the ring. This is a CH2 group, right? So there's no electronegative atoms here. There's none in alanine. This is just a methyl group hanging off, okay? There is a nitrogen here in tryptophan, but there's so much carbon and hydrogen that it's basically nonpolar. Isoleucine, no electronegative atoms in the R group here. Leucine, no electronegative atoms, and valine, no electronegative atoms. Okay, so when you build a protein, what happens is that you attach these amino acids one after the other, building peptide bonds between them, and you make this long chain of amino acids. All right, so let's explore the structure of protein.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. a hydrogen atom. And 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, enzymes are just proteins that help to speed up chemical reactions. Okay, chemical reactions are basically what make us live, And there's three ways to speed up a chemical reaction, right? You can raise the temperature, which tends to speed it up because that makes more movement happen. You can increase the concentration of the things that are reacting so that if there's more of the stuff that needs to bump into each other to make something, more bumping into happens, okay, because the reaction vessel is more crowded. And then the final way is to introduce a catalyst, which is some third party, a molecule or an element, that helps the reactants, the things that want to form the new compound, get together more easily and more quickly, while the catalyst itself is not destroyed in the chemical reaction. Okay? Catalysts can be inorganic, they can be organic. Okay? In living systems, most catalysts are called enzymes, and they are proteins that speed up the chemical reactions of life. So these are the tools of life, basically, one of the many tools of life. By lowering the activation energy of the chemical reaction, we speed the reaction up without having to do things like change the temperature of the cell or change the concentration of the reactants in the cell. If we were to do that, we would create toxic conditions in the cell that would kill it. Okay? So we want to run everything at body temperature, which is 98.6, and by throwing a catalyst like an enzyme in there, that allows this to happen without messing around with the temperature or the concentration. Okay? So let's learn a little bit about enzymes.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
When molecules collide, there must be a minimum collision energy for the reaction to occur. Lacking this energy, the molecules remain intact and no change results from the collision. The colliding molecules must have a total kinetic energy equal to or greater than the activation energy, which is the minimum amount of energy required to initiate a chemical reaction. Click on endothermic or exothermic reaction to view an animation of the energy profiles for each of these reactions. This is an energy profile for nitrogen dioxide, NO2, reacting with carbon monoxide, CO, producing nitrogen monoxide, NO, and carbon dioxide, CO2. Click on each of the regions, A, B, C, or D, as they appear to view the molecular changes that occur during each stage of this reaction. As the two molecules approach each other, an attractive force begins to form between one of the oxygens of nitrogen dioxide and the carbon of carbon monoxide, which is indicated by the dashed line. The bond between the nitrogen and oxygen of NO2 begins to weaken, and the bond length increases. When molecules collide, they form an activated complex, which is a temporary species formed by the reactant molecules as a result of the collision before they form product. In this reaction between nitrogen dioxide and carbon monoxide, the activated complex is NO2CO. The activation energy, which is the minimum energy needed to initiate this reaction, is 132 kilojoules per mole. At this point in the reaction, the bond between carbon and oxygen continues to strengthen, and the bond between nitrogen and oxygen continues to weaken. This is an energy profile for nitrogen monoxide, NO, reacting with chlorine molecules, Cl2, to produce nitrosyl chloride, NOCl, and chlorine atoms. Click on each of the regions, A, B, C, or D, as they appear, to view the molecular changes that occur during each stage of this reaction. As the two molecules approach each other, an attractive force begins to form between the nitrogen of nitrogen monoxide and one of the chlorines of the chlorine molecules. This attraction is indicated by the dashed line. The bond between the chlorines begins to weaken, and the bond length increases. When molecules collide, they form an activated complex, a temporary species formed by the reactant molecules as a result of the collision before they form the product. In this reaction between NO and Cl2, the activated complex is nitrile chloride, NoCl2. The activation energy, which is the minimum energy needed to initiate this reaction, is 85 kilojoules per mole. At this point in the reaction, the bond between nitrogen and chlorine continues to strengthen, and the bond between the two chlorines continues to weaken. A bond has now formed between nitrogen and chlorine. The bond between the two chlorines has broken. The products of this reaction are nitrosyl chloride, NoCl, and chlorine atoms. The products are less stable than the reactants. The products have a higher potential energy. In this type of reaction, heat will be absorbed from the surroundings by the reacting mixture. The heat absorbed, or delta H, is 83 kilojoules per mole. This is an endothermic reaction.
Okay. Some functional enzymes called holoenzymes have two parts. The apoenzyme is the protein part, and then sometimes there's a cofactor or a coenzyme, which are like little tools that the enzyme uses to do the work of making the chemical reaction happen. Okay. The cofactor can be a metal ion. You get that from your diet. And the coenzyme is usually a vitamin. Okay? The enzymes are always specific for their substrate, which is the thing that they act on to make the new compound. All right? And they usually end in the letters ASE, and they're named for the reactions that they make happen, like hydrolases and oxidases. Right? Oxidases do oxidation reactions. Hydrolases use water to split chemical bonds. Other examples include kinases, which add phosphates to things like proteins. There's all sorts of different enzymes that do different work, okay? They're all proteins, and they all generally have a globular shape, all right? And they have a little indentation on their surface where the business of the reaction happens, and that's called the active site. Okay, so the idea here is that the substrate, shown here as these little yellow structures, binds to the active site, and then the enzyme helps to make a new chemical bond in this case, and then it releases the compound, and then it can turn around and do the same thing again really, really fast. Okay, so the result of this is that we're able to make a lot of what the cell needs in order to live without having to spend a lot of time waiting for it to happen, just from random collisions. Okay. Our next topic is lipids. So let's learn a little bit about lipids.
Lipids are made of carbon, hydrogen, and oxygen atoms, arranged in long chains consisting mainly of carbon-hydrogen units. 3 fatty acid chains can be linked to a glycerol molecule to produce the lipid known as a triglyceride. As usual, the fact that a dehydration synthesis takes place tells you that the reverse reaction, hydrolysis, is also possible. Phospholipids are one very important class of lipid. Phospholipids make up the plasma membrane of the cell. Their function is directly tied to their structure. The head of the phospholipid is hydrophilic, which means it is attracted to water, while its tail is hydrophobic, which means it is repelled by water. When phospholipids are surrounded by water, they naturally form 2 layers so that the hydrophilic heads are always turned out toward water, and the hydrophobic tails are always turned in toward one another, where the water cannot touch them.
So, as we saw, right, triglycerides are one example of a lipid, right, where you have 3 fatty acids. Again, there's the acid group here, here, and a glycerol backbone. Glycerol is actually a 3-carbon carbohydrate. And then you form this molecule that has a polar and a nonpolar portion, right? The nonpolar portion is all this carbon and hydrogen hanging off the end, right? And the polar portion is this backbone here. And you create it by dehydration synthesis, right? You remove 3 waters, right, to make these new bonds between the glycerol and the fatty acids, right? And if you do the reverse, you use hydrolysis reaction to split these bonds and free up the fatty acids. All those carbon-hydrogen bonds mean that there's a lot of energy in lipids. Lipids are the calorie champ as a result, right? They have more chemical energy in them than carbohydrates or proteins do. And that's also why the body uses lipids for fat storage, because it takes up less volume and stores the maximum amount of calories, okay? The fewer carbon-hydrogen bonds that you have in a lipid, the less energy you're going to have. So in an unsaturated fatty acid, where double bonds are present, you can see here, there are fewer carbon-hydrogen bonds, which means that there's fewer calories, right? The other consequence of this is that because of these double bonds, you put kinks in these chains, and they don't stack real well, so they tend to be liquids at room temperature, like corn oil and safflower oil and olive oil, right? Whereas in saturated fatty acids, where the hydrocarbon tails are totally saturated with hydrogen, so they have a lot of calories in them, they stack real well, and these tend to be solids at room temperature, like margarine and lard, okay? Crisco, all right? The use of triglycerides to store energy in the body is also important because it holds in our body heat, and it pads and protects internal organs, all right? And obviously serves as a fuel storage depot, okay? The formation of fat, right? You can see here examples of saturated and unsaturated fats. The unsaturated fats, the heart-healthy fats, have those kinks in their chains, which don't stack, and that's why they're liquids at room temperature and have fewer calories. So people say you should cook with unsaturated fatty acids or unsaturated oils, right? Polyunsaturated oils, whereas saturated fatty acids are less healthy for you because of all the calories they contain. They can cause the deposition of plaque on your arteries, and that leads to hypertension and puts you at risk for a stroke or a heart attack, okay? A type of saturated fat called trans fat is used in snack foods in order to increase shelf life, all right? So it's not a plot to make you fat, it's just a plot to increase the amount of time that the snacks are good on the shelves. And so the watchword here is to limit your consumption of trans fat in order to improve your heart health and to not gain a lot of weight. Okay. Another type of lipid is cholesterol, which we get from meat and our dairy products. Okay. Cholesterol is important because it's the source material for hormones like estrogen and testosterone. Hormones are chemical signals that are produced by endocrine glands in the body that enter the bloodstream, and then once they find tissues that have cells that contain receptor proteins on their surface for those hormones, the hormone and the receptor bind, and then that causes the cell to perform an action dictated by that chemical command, which eventually creates conditions that reduce the amount of that hormone in your system, and that's called negative feedback. Okay. Cholesterol itself is also used in bile salts, which help to emulsify the fat in the food we eat, so it acts kind of like a detergent in order to make it easier for us to break down the fat in our diet. And it also aids in something called membrane fluidity, where the cholesterol gets stuck in the membrane by our cells, and that reduces the movement of the phospholipids in the membrane, which are the building blocks of most cell membranes. Okay, and a phospholipid is simply a triglyceride that instead of having, let's look here, instead of having a 3 fatty acids attached, one of these positions is occupied by a phosphate and usually some kind of an amino group. All right, and that produces a molecule that has an amphiphatic quality, meaning one end is very polar and the other end is very greasy. Okay, and in water, phospholipids in the cell membrane tend to form what we call a phospholipid bilayer, which we'll talk more about when we get to the organization of the cell. But basically it forms a semi-permeable barrier through which only certain things can move. Okay, and that helps the cell do its work. ATP, okay, is part of a group of molecules called nucleotides, which are the building blocks of DNA and RNA. Okay, ATP is the universal energy currency, and it's made up of a nitrogen-containing base, a pentose sugar. Pentose means 5, right? So there's the carbons, 1, 2, 3, 4, and 5. And then generally 3 phosphates on the end. The phosphates have negative charges, so to hold those negative charges together, the covalent bonds between them have to be very strong, and that's where most of the energy is stored. So when you crack those phosphates off in ATP, you release a lot of energy and you can do a lot of work. Okay, the base in ATP is called adenine. All right. In RNA, the building blocks are called ribonucleotides, and they have an OH at the 2-carbon position. And in DNA, there's simply a hydrogen at this 2-carbon position. Okay, so let's learn about DNA and RNA. Okay.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So, we've talked about levels of organization in organic molecules, right? Carbon is the basis of life because it can form rings and chains with itself, and carbon-containing molecules have a primary structure, which is the sequence of the building blocks in the carbon-containing compound. In proteins, those are amino acids. In RNA and DNA, they are nucleotides, and in polysaccharides, they are monosaccharides. Okay. There's secondary structure, which is basically things like helical turns and twists, and then there's tertiary structure, in which the regions of secondary structure interact with each other, and then there's quaternary structure, in which individual chains of polymers can interact with each other. Okay, and we see this to a large extent in proteins, to some extent in carbohydrates, and to some extent in nucleic acids. Okay, a little bit in lipids, not that much. All right. Inorganic molecules don't contain carbon, okay, and most of them are salts that are the source of things like electrolytes, and we discussed the importance of those in living systems as well. Okay. All right, I will see everybody in the next podcast, and I thank you for listening.